All right, good morning, everybody. Hope you can hear me just fine. If not, let me know. Welcome to EPA's Computational Toxicology Communities of Practice. My name is Cameron Clark. I'm a student contractor in NCCT. This morning we have Dr. Josh Harrill, a toxicologist in NCCT, presenting on high-throughput transcriptomics, concentration response screening, and MCF7 cells. Like I said, if y'all have any issues with audio or video, just either speak up on the line or type it into the chat box. And I will let Josh go ahead and take over here.
Okay, so should we pull these in the full screen?
Yes.
Okay. Well, thank you for the opportunity to talk here today. I'm going to be presenting some information on our high-throughput transcriptomics screening efforts in the NCCT laboratories, in a particular model, these MCF7 cells. I've presented this information a couple times earlier this month, so some aspects have changed. And given the format of the webinar, I'd ask for folks to save their questions till the end, and we'll try to save some time for discussion at the end. So first, my conflict of interest statement. I have no conflict of interest to declare. The views expressed in this presentation are my own, and even though I will talk a lot about a certain product and trade name, that does not constitute endorsement or recommendation for use. As far as the outline of what we'll be talking about today, I'll go through some background and objectives of our pilot project and our larger project. And the majority of the talk will focus on this high-throughput transcriptomics pilot experiment that we've been running over the last few months, with a particular emphasis on the optimization steps we had to use for our culture conditions, a concept called attenuation, which is something that is used with the BioSpider TempoSeq platform, and as well as some notes on our experimental layout. Then I'll get into some results, particularly pertaining to assay performance metrics and concentration response modeling that we've done on the data in a preliminary form. And then I'll move on to one slide on current activities and future directions. So just one slide on background of why we're doing this. ToxCast, as it exists right now, covers about 320 genes. And even though that's quite a few genes, that is not the entire universe of biological diversity you could expect in a cell, as far as the genes that are expressed. As far as pathway coverage, we do a little better as far as covering the biological space, with a little over 50% of known pathways covered with at least one gene within ToxCast. So really the goal of what we're trying to do here is to leverage some recent technological advances in transcriptomics that provide for rapid and cost-effective whole transcriptome screening to increase this biological space, and to eventually use this high-throughput transcriptomics platform as what we're calling a broad-based tier 0 screen for bioactivity. So casting a broad net for the activity of chemicals. As far as the technology we've chosen to use, we're using the BioSpider TempoSeq platform. We're using the TempoSeq human whole transcriptome assay. This is a targeted RNA-seq technology that provides output on greater than 20,000 transcripts. It requires a very low RNA input. Less than 10 picograms are required to achieve a whole transcriptome profile. And the instrumentation needed to perform this assay can be done on standard PCR and next-gen sequencer instruments. So it's a specialized assay platform that doesn't require particularly specialized technology. It's also compatible with both purified RNA or cell lysates that you can generate directly from cells cultured in multi-titer well formats. As far as how the TempoSeq assay works, these lines in blue, if I can find my mouse here, these lines in blue represent an mRNA transcript species. And the way their technology works is that for each targeted gene or each targeted transcript, you have a pair of detector oligos. These detector oligos anneal to the target mRNA molecule and are ligated. The ligation product is then eluted, and use PCR amplification to attach barcoded primers, which allow you to track sample identity within each transcript, as well as a universal sequence for annealing to a high-seq flow cell. Then you do standard cluster generation and sequence alignments to actually get your counts at the end. So the objectives of what we were trying to do, beginning with this technology, we wanted to run a pilot study where the goal was to optimize culture and assay conditions for high-throughput transcriptomic screening in MCF7 cells as our chosen cell model, using this TempoSeq whole transcriptome assay. We wanted to evaluate TempoSeq assay performance, determine the ability of the TempoSeq assay to detect known biological signatures following chemical perturbation with a small set of 44 chemicals in concentration response, and then use this data to try to guide further experimental design of larger screening studies. So this is a table that summarizes our experimental design. Once again, we're using MCF7 cells. For this pilot study, we looked at two different culture conditions, a standard media, which is DMEM plus 10% heat-inactivated fetal bovine serum, as well as a secondary media condition, which is phenol red-free, PRF, PRF media with 10% charcoal strip FBS. The reason we wanted to do this is we know that there's an unknown complement of factors in any type of standard serum, which can activate estrogen receptor and also activate many other things. So we wanted to use this data to evaluate whether we wanted to try to have a more defined serum condition within our culture and maybe, I don't know, reduce the noise in the culture system by using a different media. Turns out that was an interesting idea, but it didn't turn out the way we thought. As far as time points, we're using 3 different time points in this study. Time points meaning exposure durations, a 6-hour exposure duration, a 12, and a 24-hour exposure duration. And we're actually using 3 assay formats. We're using the TempoSeq whole transcriptome assay, but also ran high-content imaging-based apoptosis and cytotoxicity assays in parallel with the TempoSeq experiment. We did a 8-point concentration response with half-log unit spacing and ran 3 biological replicates, 3 of which we sent for sequencing and 1 we held in reserve here at NCCT. As far as the pilot workflow, this is what this looks like from the laboratory perspective. The first thing we did was generate some cryopreserved stocks of MCF7 cells that we purchased from ATCC. We developed a standardized expansion protocol, so every time we run a biological replicate within these experiments, we're taking cells out of the freezer and taking them through a uniform expansion procedure, so we're always using the exact same passage number for each experiment. We then use some laboratory robotics to perform cell plating and actually dosing of the cells, which I will elaborate on here in a minute. And then this diagram over here to the right just shows our different assay tracks as far as the targeted RNA-seq and the cytotox. So next, I'll talk a little bit about our optimization. And we had to look at a lot of different parameters for optimizing this assay before we felt good actually running this pilot experiment. For the actual cell culture itself, we took the time to authenticate our cells to make sure that they were in fact MCF7 cells. Like I said, we optimized our expansion protocol. We looked at different media formulations and also did some seeding density optimization. I won't talk about all of these today, but I will touch on a couple. What I'll focus on is the optimization steps we had to take for our TempoSeq assay as far as lysis conditions and attenuation of highly expressed genes. And then for chemical treatments, we're doing some rather novel things regarding randomization of our treatment plates that I'll get into here in a moment. Just briefly looking at our MCF7 expansion protocol. So for every biological replicate we do, we pull a fresh set of MCF7 cryostocks out of the freezer. We thaw them and take them through 3 serial passages through ever-increasing sizes of culture vessels. So we start with a little T25 centimeter square flask and expand them out to a larger T225. And after the T225 passage, then we passage into test plates. So we're using this uniform procedure every single time, and the way we have things set up, we can actually do serial thawings on consecutive days and serial sampling on consecutive days. So when we're actually running a screen, it's in a very compacted time frame. Like I said, we authenticated our cells both by STR profiling and karyotyping prior to use in our studies here. This is a little bit of elaboration on why we're looking at these 2 different media formulations. So the images here are MCF7 cells in culture, in 384 well culture, stained with Alexa 488 phalloidin to look at their cytoskeletons, and basically Hertz stain to actually visualize their nuclei in blue. And what we wanted to look at was both not only the rate at which these cells proliferate, but also how fast they reach confluency. And we wanted to try to keep our cultures at subconfluent levels at the time of sampling in each exposure duration that we had. We had some interesting qualitative observations from this. We seem to get more cell attachment and less cell spreading, or more cell attachment and more cell spreading with this phenol red-free charcoal stripped media, but in a greater increase in cell confluency over time. The cells seemed more efficient in attachment and then just spread it out, forming a larger cytoplasmic area. As far as our normal media, we saw good attachment, however slightly less efficient, but we also saw the propensity for some of the cells to actually form these little ductiles and detach from the plate. So these cells also grow faster in this normal media as compared to here. So I won't belabor this anymore to say there's even qualitative differences you can see with your eyes between these two culture conditions, and we move forward with both of them in the pilot study. So now onto this topic of attenuation. So for those unfamiliar with this, attenuation is a method that can be used with the BioSpider TempoSeq assay to prevent highly expressed genes from occupying a disproportionate amount of the available read space in any sample, and therefore increase the ability to quantify low abundance transcripts with greater precision. So attenuation itself is accomplished by adding what we call cold probes. This is a term we came up with ourselves. These cold probes do not have the PCR amplification tags at the 5' to 3' ends of the detector oligos. So what will happen is these red and green sections on the attenuation probes are not present. So those attenuation oligos will actually anneal to the mRNA species that they're targeted against and prevent this ligation step and subsequent PCR amplification. So you're basically using a stoichiometric process to remove a number of transcripts for any given species from the amplification pool. And that's how you're actually attenuating or artificially lowering the abundance of those transcripts. There is a standard attenuation that BioSpider performs for ribosomal RNAs that's applied to all TempoSeq whole transcriptome assays. At least that's my understanding. So for additional attenuation, if you wanted to look at your specific model, you have to answer a couple of questions. The end user must define what sets of genes on top of these ribosomal RNAs would you like to attenuate and what degree of attenuation is appropriate. So as you can imagine, in a cell model that's novel to you, this is a rather complicated question to try to figure out. So that's what we needed to do. We needed to answer the question within our optimization steps, is additional attenuation needed for our MCF7 cell model to increase signal? And if so, how are we going to define that set? And so we set up an experiment to do this. And the way this experiment was set up is we had 3 components, which you see outlined here in this yellow, blue, and green. With each component, there's different treatment conditions. So we did an untreated time course in both of our media formulations where we created cell lysates at 30, 36, and 48 hours after plating, which correspond to the exposure durations we were using given a 24-hour attachment period. We also used DMSO, which is a vehicle control, and an HDAC inhibitor, trichostatin A, in each media formulation at 2 different exposure timings. So from 24 to 30 hours and 30 to 36 hours, we had these 2 conditions. HDAC inhibitors tend to increase the expression of a whole lot of things. So what we were trying to do with this experimental design is identify genes which were consistently highly expressed no matter what we did to the cells. Okay? So contained within this whole design is 14 individual conditions and 42 samples. And what you'll see over here on the top left is a log2 read count distribution for all 42 samples in this experimental design. So even though these lines are superimposed over each other, each line corresponds to a sample. And what you can tell from this is about half of our probes out of the 21,000 we looked at had a 0 read count. So that can be expected. Not all genes are expressed in all cells. So we have about half of our 21,000 probes had a non-zero read count. And then you see this broad range in the actual abundance of transcripts ranging from 1 all the way up to several tens of thousands for any particular gene. So it's a very broad range. So another way to look at this data is if you plot a cumulative distribution, which is what we've done down here on the bottom. So each one of these curves, once again, is an individual sample. And we wanted to ask the question, how many genes does it take on a per sample basis to account for 50% of our read space? And the answer is not that many. So out of 21,000 potential probes, between 200 and 300 actually occupy 50% of the read space on an individual sample level, which leaves the other 50% for the other 9,000 probes that you have. So that really limits your ability within that other 50% to look at low abundance transcripts without some additional attenuation. So the next question we had to have, given this observation, we can answer the first question, yes, we felt that we needed to attenuate some additional genes in these samples. So how do we go about identifying our attenuation set? So what we came up with is this concept of a commonality analysis, or commonality score. So basically what we looked at is the overlap. We established a candidate list for all 14 of our test conditions, and then basically looked at the overlap of those lists for the genes that were in the top 50% of the cumulative, or the bottom 50% rather, of the cumulative distributions for each condition. And what we determined is if you have a gene here that has a commonality score of 14, which is the 14 conditions in our study, you can interpret that as a gene that's consistently highly expressed regardless of perturbation. So that would be a good candidate for attenuation. And when we ran through this process at the end of the day, what we were left with is about 156 detector oligos identified as candidates for attenuation in our NCF cell model. I'll say here that we did this in cooperation with BioSpider, but we took the tack where we set a goal where we wanted to reclaim about 50% of our read space. And we asked the scientists at BioSpider, give us your strategy. You go over here, do your strategy. We'll go over here and get our strategy. Then we'll compare them at the end and figure out how close they were. And the overlap between the sets we came up with was about 95%. So this is a fairly consistent way to do this, even though I think this is the first time this has really been done. As far as the actual set we have, so this is our 156 probes. We plotted our log2 read counts as these box plots. As you can see, there's a gradient of expression, as you'd expect across the many different genes. And what the goal is for attenuation and the guidance we received from the BioSpider scientists is that you do not want to attenuate below a residual read count of 100. So even though these read counts are in the several thousands here, you don't want to drop this to the point where you run the risk of attenuating so much that you've actually eliminated the expression of that gene altogether. So the game here was to lower all these right around to the residual read count of 100 mark. And even though I don't have the data in this presentation, we were successful in doing that. We can see a clear performance increase or consistent decrease in the expression of these genes in an attenuated versus a non-attenuated assay. And what the actual practical consequence of this was for us in terms of our study is that on this plot here shows that we were actually able to regain between about 35 and 45% of the available read space using attenuation versus non-attenuation. And so what that means is that if we request a target read depth of 3 million for a sample and we use an attenuated assay, the data we get back are similar in quality to if we ran it at 5 or 6 million. So you're actually increasing your resolution at the same, I'll say, price point at this point. So it's really, really a way to focus in on these lowly expressed genes in a more efficient manner. The alternative being increasing your read depth. Okay. And as you'd expect, the most highly expressed genes in this attenuation set are in fact housekeeping genes, cytoskeletal genes, ribosomal genes, other genes like that. So I'll switch gears a little bit at this point and talk about our chemical test set and the way we apply chemicals to our plate. So this is the list of the 44 chemicals we tested in this pilot study. The chemical set covers a broad range of mechanistic diversity, but also has redundancy within mechanistic class. So we have a little over a dozen different MIE families here, and each one has between 2 and 4 members. And I'm happy to circulate this list to whoever's interested. As far as how we determined our dose range, we looked at data from Judson et al. 2016 to look at what the kind of the lower boundary of cytotoxicity was for a lot of these chemicals, or what the distribution of cytotoxicity values may be for these chemicals as determined from in vitro DV. And what we found is that in most cases, you start to see cytotoxicity with these chemicals below 100 micromolar. So what we did, we just set our upper threshold at 100 micromolar and then tested concentrations below that. So we used a uniform concentration range of 0.03 to about 100 micromolar for each of these chemicals. As far as our dose plate layout, so for this particular experiment, given all the time and media conditions that we have a total of 18 plates in play across 3 biological replicates. And we use the same dosing plate to dose every single one of those 18 test plates. And this is the layout of the dosing plate we used. There's several features here. The columns in green on the top and bottom half of this plate represent our individual test chemicals and the actual test concentrations that we used. The chemical plates themselves were prepared as 200x stocks of these values. Over here in this column, you can see we have some wells designated as non-treated control. So there'd be 3 wells in every test plate that didn't receive any treatment whatsoever. We have 4 DMSO controls. And we have these chemicals in red, these 3 chemicals, which we're referring to as connectivity map reference chemicals. So one of the reasons we selected MCF7 cells is the ability to compare back to the Broad's connectivity map database. And these chemicals are highly represented within that database and allow us to evaluate whether we see accurate biological signatures in our cell models given the treatments that we apply. So each of these reference chemicals are applied to the test plates in triplicate, while all the test sample conditions are in singlet. And then we have 4 DMSO controls. Over here, we have our high-content imaging cytotox and apoptosis positive and negative controls. So those are just chemicals we use to evaluate the performance of our HCI assays in-house. Okay. So the way we actually distribute the chemicals from this single dosing plate is using an acoustic dispenser, the LabSite Echo 550. And the way this dispensing technology works is that it actually uses sound waves to precisely transfer small precise quantities of liquid, 2.5 nanoliters, from a source plate that's right side up in a loading tray within the instrument to the test plate, which is actually hanging upside down over the source plate. So you're basically shooting these little droplets of liquid through the air to actually get to actually dose your plates. But one of the reasons we wanted to use this, besides the demonstrated accuracy of this method for delivering volumes of liquid, was the ability to randomize our test conditions within a plate. So one of the traditional problems in vitro testing is that you get edge effects on a lot of different assays, and if you systematically put your treatments in the same place, you're going to get this systematic artifacts in your data. So to guard against that and to allow us to use the entire real estate of our test plates, we came up with a scheme where we uniquely randomize every single test plate and then deconvolute the data on the back end. Okay. And so this is how this looks like from the scheme here. We have our single dose plate. Here's our assay stream 1 for HTTr. And you can see all the greens and the reds, our controls and our reference and our treatment samples are randomly applied to the plate. And we leave this first column open for the TempoSeq assay in order for us to be able to manually pipette in some QC control samples. These QC control samples are the same ones that were used by the Microarray Quality Control Consortium group and allow us to track assay performance from plate to plate and from run to run over time. Those are purified RNA products, so we also generated some bulk lysate, DMSO and trichostatin samples that we use in every single plate that we run, so we can track the performance of lysates across time and across runs. We also have lysis buffer blanks, which basically you can equate to an assay blank that allow us to see if there was any type of contaminant in the lysis buffer we were using to create these lysates. We leave the bottom 6 wells in this first column open, as BioSpider, when they receive the plates for processing, add their own MAQC and B samples and their own lysis buffer samples so we can evaluate whether there was any change during shipping which may have affected the performance of the plate. So we compare ours versus theirs for MAQC. And for high content imaging, we do the same here. We randomize every single plate except the blue is also distributed throughout. And then we have these no dye label controls, which allow me to look at the effectiveness of our dyes against background in these HCI assays. Now we'll talk a little bit. We've sent our sample to BioSpider. We've gotten the data back. Now what do? So this is our strawman data analysis pipeline. The way this works is we eventually will be taking FASTQ files and generating our own read counts. Right now we use the read count files generated by BioSpider's alignments. We filter for a minimum mapped read depth, and so toss out samples if they don't meet a minimum mapped read depth of about 300,000 counts. Those that get kicked out become candidates for resequencing, and if they come back with a better sequencing depth, we can include them, and if not, we just exclude them all outright. We then subset the data by chemical, media, and time, including the corresponding DMSO controls within each condition, and filter that data, these parsed packages of data, by flooring the data using a median raw read count of greater than or equal to 5. So we're getting rid of a lot of probes that just have no expression or very unreliable expression. We then move that data, that floored data, to the DE-seq2 package, which is becoming an industry standard package for RNA-seq analysis to generate, to use the shrinkage estimates and other mathematical operations within there that I won't try to explain here, to generate estimates of fold change from control along with estimates of standard error and a p-value that we can then aggregate at the gene level and use for MIE and MLA identification. We also do benchmark dose modeling at the gene level and use that information for pathway enrichment type studies. This is still a draft pipeline, but this is what we've established so far with this pilot data, and we're still working on it. So as far as assay performance metrics, what we can look at from this data is this concept of total mapped reads versus percent mapped reads. And this is what we found as the most useful metric for us in determining the quality of a sample or the performance of a plate. So RNA-seq data, the way that you can roughly equate the total reads that you get as the cluster density for a particular sample within a flow cell. You then take the sequences that are generated from that flow cell and you map them against a reference genome. In this case, it's BioSpider sequence probe manifest. And based on a certain stringency that you apply, you can then score each read as either mapped or unmapped. So total reads equals total mapped reads versus total unmapped reads. And from that, you can calculate a percentage. So looking at that, we can tell which sample didn't have a very high read count and also didn't do particularly well when mapping to the reference genome. For performance, we also looked at correlation and variation within technical replicates. So those are the DMSO controls and the reference chemicals that are contained within each plate, an N of 3 or 4. And then we looked at correlation and variation in biological replicates across plates. So that's our individual cultures, which constitute our biological replicates. Then we went through some exercises trying to detect biological signal with chemicals that we know what they should do within our test set. And I'll get into a little bit of that later on. So this is a plot that we found particularly useful as a diagnostic plot. It's percent mapped reads on the x-axis versus total mapped reads on the y. Okay? And what this plot is, we've color-coded each of our different quality control or treatment samples by different colors. Our test samples from the study are in blue. And we've just plotted this out to see different aspects of the data. What I can tell you is that our average total mapped reads from our test sample population is about 3 million. So that's what we asked for. We needed a mapped reads of about 3 million. And our average mapped read count per gene is 150. Our percent mapped reads for a majority of these test samples are above 75%. So that's the threshold of about right here. The lysis buffer blanks, both ours and the ones they use at BioSpider, have very low read counts. They're not 0. Some people may expect that value to be 0, but in fact, we've done some work with the folks at BioSpider to actually determine the nature of why we're getting some residual reads or some residual noise in these lysis buffer blanks, no matter who's doing it every time. And this is not something unique to our group. What I can tell you is that across samples, the identity of the counts in these lysis buffer blanks is not consistent. It seems to be random noise in some fashion. And what we also, so they're down here in the red. What we also discovered is that purified RNAs, our MAQCAs and Bs, tended to have a higher mapped percentage and higher read counts than our test samples. So it seems that purified RNAs do perform a little bit better as far as mapping to the reference genome than cell lysates do. But you still get fairly good quality data from cell lysates. The other thing we saw was what I'm calling this comet tail, which will become apparent what this is in a moment, and this offset cluster that's down here that has similar mapping rates to a majority of our test samples, but just low total reads. Okay. And so to discover what some of these things are, we can look at the same plots broken out by plate. And so obviously this comet tail here is attributable to a single plate that seems to be a poor performer. So this is one that we've sequenced a couple of times now and eventually intend to exclude from the study just a poor performance of unknown origin at this point. All the rest of the samples seem to cluster very tightly up here above the 1 million read mark and above the 75% mapped read mark. These clusters here, circled in red, were the candidates for resequencing. We actually have resequenced these and saw them move up into the main body of these point clouds. So those were some kind of technical failure on the first time around, which didn't repeat the second time. So we were able to rescue those samples. So as far as correlation among technical replicates, I don't want to belabor these next few slides, given they're kind of difficult to read, but what we looked at was the pairwise correlation among like technical replicates on each plate, and just plotted the correlation coefficients here for each of our different control sample types. Basically, the take-home message here is that on the read count level, the raw read count level, our correlation among technical replicates is very high. It's greater than 0.85%. So we're seeing good replicability across across samples of like type within a plate. You can see here there's some points here in this particular condition, DMEM24, that were low. That's our poor performing plate. So that's what's causing these points to be lower on the graph. As far as coefficient of variation among technical replicates, our CVs and gene expression values is low, with a median less than about 30%. So that's comparable to what you'd see on a well-performed bathymetric study. So these graphs are of the same nature. We've plotted distributions of CV values at the gene level for each of our conditions, and each small distribution within a type was a particular plate. Okay, so take-home message here: our coefficients of variation are very low. Our correlations are very high at the technical replicate level. As far as correlations in biological replicates, we looked at this to make the data a little clearer. We stratified it by this shouldn't say expression level, this should say dose level. So regardless of chemical, we stratified these box plots by dose level just to see if we could see an increase in the correlations of either our raw counts or our log2 fold changes as a function of dose, because you would expect the biological signal to start to separate itself from control at higher doses and thus get higher correlations, perhaps. And that's what we see. We see some trends like that, where we start here with pretty good correlation even at the lower concentrations for log2 fold change of differentially expressed genes. That starts to go up in these tails by a modest amount. And so what the take-home message here is, once again, across our biological replicates, so this is across different cultures, we see a high degree of correlation in our read count values and our fold change estimates for like conditions. As far as our coefficients of variation, once again, they are low. Here we see a trend that has been shown in the RNA-seq literature previously for DMSO and trichostatin, is that as you increase the mean read count for a gene, your coefficient of variation decreases. So when we get into an area of this point cloud here, where we start to have reliable abundance of transcripts, you can see the CV starting to decrease. And once again, they're around 30%. flatten out. So this is not bad. We also stratified these graphs. These are media by time conditions again, but stratified it by the level of transcript abundance for different bins going from low to high, and plotted the distribution of median CVs and the distribution of 90th percentile CVs from like conditions. And once again, we see that, around 30% or a little under 50% for these low abundance genes, and then that decreases as you increase the absolute abundance of these transcripts. So it's a very good coefficient of variation and replicability among these samples. So we took that as good news for replicability and assay performance. The next thing we wanted to do is try to pull some biologically meaningful signal out of this data. One of the things we tried is to look at this estrogen receptor alpha biomarker signature that was published in Ryan et al. 2016. So to construct this biomarker, they looked at many different estrogen receptor agonists and antagonists and built a consensus gene set that came with some type of aggregate of fold change and directionality that goes with it. So you can see here for agonists, the ER biomarker, these genes go up, these genes go down. For antagonists, it's the other direction. These genes go down and these genes go up. So it gives some directionality in what we're seeing. Contained within our chemical set, we had, I believe, 4 estrogenic chemicals and 3 anti-estrogenic chemicals. And so basically what we did is take this biomarker profile and did some correlations of our own just to see if we could see the type of correlations and directionality that we expected based on this biomarker signature. So here is the chart of our chemicals that were anti-estrogenic or estrogenic, and these numbers correspond to our columns here. Each of these are our different media by time conditions, and these are stratified by doses here. And what we can see is that if you have a negative correlation with the biomarker, you should be seeing an antagonist, and that's what we're seeing here. All of our antagonists, for the most part, produced a negative correlation in this biomarker, with some conditions, media time combinations, giving stronger correlations than others. With the estrogenic compounds, it was the opposite. If positive correlations are in blue, and so that's what we see here with our estrogenic chemicals. In most every condition, we do see positive correlations with the biomarker set. The performance in DMEM24 is somewhat limited by the plate we have not discarded at this time from this analysis, so this includes the bad plate. So this may improve if we actually got rid of that or excluded that data. But another thing that we saw that was interesting just doing this type of analysis is here within the phenol red free strip conditions. If you look at conditions 2 and 3, those correspond to our selective estrogen receptor modulating chemicals. So these are a specific type of estrogen receptor antagonist. And what we saw is when we used the stripped media, we actually decreased our ability to detect these, or to detect the biological signal associated with these. However, we did not compromise the ability to see a selective estrogen receptor degrader. So if you're taking this as part of your decision logic for which media to pick for a larger screen, we would favor the DMEM over the stripped media, given it gives us the ability to see, at least for this particular receptor, a larger variety of biological mechanism. And as far as the correlation with our ER-alpha transcriptional biomarker, what we did for these plots, agonists are on the left, antagonists are on the right, and we basically scored each chemical as being an estrogen receptor agonist or not, or an estrogen receptor antagonist or not, and plotted the distribution of correlation coefficients with the biomarker. And what you can see is that for estrogenic compounds here, which are in the blue, they clearly separate in terms of correlation coefficients from the not ER agonist population, and likewise for the antagonists. So clear separation there when you score them in that manner. Although you can even see in this plot that with the stripped media, your ability to detect these selective estrogen receptor modulators may be compromised. These distributions just aren't as crisp. Another thing that we're trying, and Dr. Imran Shah is leading the charge on this, is connectivity mapping, trying to look at predict mechanism from our gene expression profiles. And so what we have here is just a very simple example of some of our reference chemicals where we tried this. We have our trichostatin A HDAC inhibitor here, sirolimus, which affects PI3 kinase and mTOR, and genistein, which is estrogen receptor. And clearly you can see these are plots of log2 fold change versus p-value. So you can see trichostatin is what we're calling our sledgehammer chemical. It just causes a lot of massive change in a lot of different genes, whereas we're seeing more subtle effects with sirolimus and genistein. When we actually tried to use the connectivity mapping approach, where we've taken the Broad database, or at least a part of it, and annotated it from a chemical-centric to MIE-centric annotation, what you can see is that our HDAC signature for trichostatin is clearly there. These spikes indicating spikes further out from the radial plot, meaning a higher signal. But we also see a whole lot of other things. Likewise, for sirolimus, our PI3 kinase and mTOR signals are there, as expected, but we also see a lot of other things. And our estrogen receptor signal is there. Once again, a lot of other things are also happening. So what we took this as is that at least for this first pass approach, we observed a large degree of promiscuity for the predicted targets that we get from any profile. And so we're currently evaluating methods to make this a little more specific and less noisy, but think this still holds some promise moving forward. And Dr. Shaw would certainly be the one to talk to about this approach. So as far as the next and the final phase that we have in this presentation, we did some benchmark dose modeling on this data, and I'll put this disclaimer out here right now. This is exploratory. This is not final by any means, but I wanted to show some interesting aspects of the data we generated. The tool we're using is the BMD Express 2.0 tool, which is being developed by SciOM under the direction of the National Toxicology Program, but also in collaboration with some folks here at EPA and with Health Canada. This is a graphical user interface tool. It was originally developed by Rusty Thomas back at the Hamner Institutes, but has since been adopted and upgraded to where it's a bit more user-friendly and contains a lot more features for doing concentration response modeling. So when you're doing benchmark dose modeling, what I've learned from my experience with this is it really is a game of choices. There's a lot of different things you can do for benchmark dose modeling, and a lot of different parameters you can set that will potentially affect the output that you get. And so the idea here is to try to find our standard approach and try to stick with it. But for right now, I'm just showing the different decisions I've put in place for this particular analysis as we're evaluating how these different choices affect our output. So from right now, we're applying a pre-filter on the chemical level, looking at the discrete chemical set within a median time condition. We're asking, are we seeing a significant effective dose using a one-way ANOVA with an unadjusted p-value less than 0.05 and an absolute fold change greater than 2 in any concentration within our concentration response range. Once we apply that pre-filter, you end up with a set of probes. We then fit, for this particular analysis, I fit 2 models, a Hill model and an exponential model, whereas you can also apply other models within BMD Express 2.0, poly-twos, powers, linear models, other things like that. But for this particular analysis, we focused on these 2. Set a BMR factor of 10%, so a benchmark response factor, about 10% change from control, and used the AIC, lowest AIC, as our selection criteria. There's features within this program for flagging of Hill models that give you an unreasonably low BMD. And so what I've chosen to do there is we've set anything that gives a benchmark dose that's lower than 1/3 of our lowest tested dose, we flag it. So we curve flag that. But I retained those values just to demonstrate a particular point within this data set. So we're going to see some BMDs that are outrageously low that are probably not real, but I kept them in there just to demonstrate something to the audience. As far as pathway analysis, that's the next step you can do within BMD Express 2.0. You can set criteria for how you want to do your pathway enrichment. Here we set our decision rules were to call a pathway enriched. Has to have 3 genes whose benchmark dose is less than the highest dose we tested. It has to have the genes that have a BMD have to comprise at least 5% of the gene set they're being compared against. And then we used the Fisher's exact two-tailed test with a p less than 0.05 as an additional gate. So it had to meet all three of these criteria to be a pathway call. The gene set collections we queried the molecular signature database curated collection, which is a collection of gene sets from online pathway databases and publications and other domain experts. Got about nearly 5,000 pathways in there. We also looked at the molecular signature database hallmark pathways. There's about 50 pathways that are coherently expressed signatures derived by aggregating across molecular signatures database. And then we also looked at Reactome, which is an open source, curated and peer-reviewed pathway database that incorporates a hierarchical pathway relationship in specific domains of biology. And some of those pathways are actually included in the MySigDB C2 set, so there's some redundancy there. Okay, so moving on to the next slide. This is what I wanted to illustrate by including the flagged hill model. So what we've got here are box plots for each of our chemical. The best benchmark dose is what each of the points in the distributions represent, and I've ranked them by increasing potency here. And what you can see is that a majority of these chemicals, we're getting distributions of BMDs within the middle half of the dose range we tested. But we do see some cases, like this chemical fulvestrant here, where with these particular rules we see an overabundance of flagged hill models. So an overabundance of curves that have a really, really low BMD. And so what that could possibly mean is that for all intents and purposes here, like for this estrogen responsive gene, MYBL1, you've actually saturated your signal already at the lowest dose. So your controls are way up here, and all the other doses are way down here, and so it tries to fit an unreasonable curve to this data. Okay, so this is still biologically meaningful information. We don't see, since we're using a common set of controls, we don't see this pattern across all of our chemicals. We only see this as really potent estrogen receptor antagonists. So we want to retain that information, but the benchmark dose estimates you would obtain from something like that are just not accurate. So we're still discussing ways on how to wrangle that data and maintain it to look at, to make sure that it's biologically meaningful and incorporated into our analysis, but just find the best way to handle it. So I've compared the same gene here for clomiphene citrate, which is another estrogen receptor antagonist, and you can see that the curve, it gives a much more reasonable concentration response curve for that particular chemical. But once again, just a range of different BMD potencies across our chemical set. If you zoom in, so now I've zoomed this box plot in to be bounded by the actual dose range that we tested, you can see the differences in potency at the probe level a little clearer. So clearly there are chemicals that are more active or less active within our MCF7 cell model. I've highlighted here bisphenol A and bisphenol B to show a particular feature of this data, and that the distributions of benchmark doses that you get for highly related chemicals within a class can be quite different. We're seeing what looks like some kind of bimodality here with bisphenol A. We're seeing some probes light up at a very low dose versus here, which seems to be a more normal-like distribution for bisphenol B. This plot here is our median benchmark dose versus the number of genes which we could assign a benchmark dose given the Hill and exponential models. And although I haven't ran a quantitative correlation on this, it's fairly clear that there's no real relationship between the number of probes affecting the actual potency of the chemical. We need to vet that statement a little harder to make that conclusive, but it was just an interesting first look at that data. I'm going to skip this table because the values are inaccurate. Suffice to say that the molecular signature database C2 gave us the greatest number of pathways enriched compared to the other more focused gene sets. But once again, these values are wrong and will be corrected in the slides that we post on the internet. All right, I'm kind of running short on time, so I'm going to try to zip through these last slides. So one of the visualizations that BMD Express 2.0 gives you are these accumulation plots, and this is a way you can look at potencies for pathway enrichment. So each curve is a chemical, each spot is an enriched pathway. The x-axis is your BMD median for the pathway, and then the y-axis is your count. So this is a cumulative plot that goes all the way up. For example, Xyram, we had about 800 pathways total affected. And so as you move across this, it's rank ordered by potency as evaluated by median BMD. And basically what I wanted to show here is just we're seeing a broad range. I've just demonstrated a broad range of probe level potencies with the BMDs. Here, as we roll this up into pathways, we see a broad range of pathway level potency estimates as well. If we're looking particularly at ER agonists or ER antagonists, we can also see a diversity not only in the relative potency of these chemicals, but also in when certain things start turning on. And by no means would I associate all of the pathways within this plot with estrogen receptor activity. But it does give you an overall broad view of when you can start to see bioactivity thresholds for the chemicals of interest that you're testing. Fulvestrant looks really weird because that's the one that we had all these unreasonably low benchmark doses. So that's why you see it stacked up vertically and then shoots over this way. But still an active chemical. So I just wanted to demonstrate this within a couple of chemical classes to show that we're moving to the point where we may be able to look at pathway level points of departure for the chemicals within our set. Another thing you can do in the last couple minutes with this data is network mapping. So what I've shown here, this is a diagram of the reactome pathway hierarchy. So when I was talking about different domains of biology, each of these central nodes is a different domain of pathway biology. And as you branch out, the pathways within here get more and more specific. They contain less and less genes. Okay, but they're related to each other in this type of structure. And what you can do, Reactome actually has a pretty nice online tool where you can query a potential chemical, look for the enriched pathways, and then map it onto these biological networks to see what type of biological effects may be affected. So for clomiphene citrate in particular, we see effects on gene expression, metabolism, cell cycle, chromatin aggregation, immune system, just a variety of different things across these different domains. So this is a good global level view of what's going on with this particular chemical. If you were to drill down into this across chemicals, you can actually use this type of structure to look at heterogeneity within a biological domain, but which chemicals may have slightly different effects. So in this particular case for clomiphene citrate, we see this branch associated with chromosome maintenance, highly enriched, and M phase, highly enriched here, whereas for another chemical, for nonylphenol, we see areas of G0 and early G1 cell cycle pathways lit up. Here, for cyclohexamide, G2M cell cycle checkpoints. So this may be a good way to leap across chemicals and kind of focus on what pieces of biology you think may be important for that particular chemical. We also can do this with signal transduction. Just another example, here's a RAF MAP kinase spur for 4-nonylphenol, MAP1, MAP3 kinase signaling, so on and so forth. So just another exploratory tool that may be in your toolbox. So as far as current and future activities for the fall 2017, we're refining our data analysis pipeline and BMD modeling approach. We're exploring additional methods for MIE prediction and characterization of biological responses, trying to prep an initial publication, and then the real effort on my end, being the laboratory lead on this work, is conducting concentration response screening of 2,200 chemicals using this whole transcriptome assay. So we're generating a lot of data in concentration response using the 6-hour exposure in our DME and media, and we're about halfway through that effort at this point. Beyond 2017, we have a partnership through Tox21. It's a reference chemical partner project where we're going to be screening more pharmacological type chemicals through our MCF7 cells to get some more defined mechanistic signatures. We plan on, of course, screening additional cell lines, and we're working on coupling with this with some image-based phenotypic screening approaches as well. Acknowledgements, this work would not have been possible without support from many different people in the center and support from BioSpider. So particularly want to acknowledge Clinton Willis. He is the heavy lifter on a lot of the culture work and has been doing an awesome job helping me get that done. Daniel Suarez, who's helped me with a lot of coordination for this. Chad Deisenroth, who helped expand the cells and took care of them before I arrived here in NCCT. And as well as our computational team, Imran, Woody, Derek, Matt Martin, who since left us for greener pastures of Pfizer, Richard and Rusty for really putting the helping design these experiments, and especially Woody and Imran for doing a lot of the analysis, and then the BioSpider team for their help and support. Looks like we got about 5 minutes for questions. So.
Yeah, thank you so much. So it looks like we've already got a question or maybe a comment on the comment box here. So Barbara Wetmore asks, you seem to have focused in on the 6-hour time point. Can you discuss in general what you've observed across the 3 time points originally proposed?
Okay, thanks for the question, Barbara. We have looked at, we're starting to look at these effects across time. We've run each of our time by media combinations through an initial run of our pipeline, and we're starting to move into actually looking at how things change across time as one of our next project goals. What I can say, just looking at the BMDs from the part of the analysis I've been doing, is that we see general consistency in the potency of a chemical across time, but we don't necessarily see similar pathways lining up at different time points. So we're still in the baby steps of evaluating why that may be or what's really going on there.
Okay, great. I think the next question we have is, are there plans to use these gene signature responses to create or inform more toxicology-relevant gene sets that could be used for enrichment analyses outside of MSigDB and Reactome, et cetera?
Thanks, Julia, for that question. That's not something we've discussed explicitly. That's certainly something that we may be able to do with the large 2,200 chemical data set. Right now, we just really haven't broached that subject internally.
All right, I think we've got a couple minutes if there's any more questions on the chat box or in the room, on the phone line. All right, well, thank you all so much for joining, and thank you, Josh. If anybody has any follow-up questions later on, feel free to reach out to me, and I can pass those on to Josh. But I hope everybody has a good day.
All right, thank you.
The leader has disconnected. The conference will be terminated.